Indeed. Yes. So I take it you can hear me okay, right? It's not a huge room, I guess. So, yeah, as Gregory says, I still work on quite a number, probably too many topics related to reproduction, but they are all related to reproduction, and even although this is largely about food intake, of course, it's all about the problem you probably well know, the broiler breeder paradox, where we have to control food intake to ensure successful reproduction. And maybe, in some respects, this topic is probably one that we have made least impression in the industry. I have quite a lot of work on osteoporosis in laying hens, and we are definitely have progress there of sorts. Egg quality, cuticle quality, and we've delivered things to the industry. But this, I feel this area is the one where we have probably had made, ironically, least impact. And so you might ask, why did I want to speak about that? But I think it's useful, A, to try and get more ideas, and we really need to think about this problem for the future of the meat industry. So, yeah, these are Scotch pies. Who ate all the pies is a saying, I guess, is if you go somewhere and find there's no food left, you'll have somebody who's maybe slightly larger appetite than the rest of you has eaten them all. But perhaps more prosaically, I could have called it the cholecystine receptor A gene underlying the largest QTL for growth in chickens, and what it's taught us about the central control of metabolism. But actually, before we get on to those genetic aspects of the control of feed intake, I wanted to look at really what has motivated us to return to this area or start in this area of the control of feed intake in poultry. And as I say, it does come down to reproduction. I'm going to look at a number of experiments we've done to look at the response of the hypothalamic feeding center, the area in the brain that controls ingestive behavior, but rather the whole metabolism, and then get on to some of the lessons we've kind of learned from genetics, and as I say, where it might be very handy to get more ideas, and that's one of the reasons I wanted to come here, where we go with broiler breeder welfare in the future. So, as some of you I'm sure know, there's something that was coined, the broiler breeder paradox. I think it was maybe coined by Eddie de Kuiper at Leuven. We have the situation where, conservatively, poultry represents at least a third of the world's meat production. This is now out of date. This was from the National Geographic based on FAO estimates, and I think I looked at the last estimate. I think it was either 60 or 74 billion chickens are killed every year for food, and with ducks next, and turkeys not a million miles behind. But they absolutely dwarf the production of animals, meat in the world, numerically at least. And the problem is, to get that number of chickens, you need that number of eggs. Every chicken obviously comes from an egg. And if birds are ad libitum fed, you have this follicular hierarchy that looks great, but does not result in mature, fertile eggs. If you restrict the birds, the follicle looks like that, and that's how it should look. So body weight and food intake control is absolutely critical to the whole industry. I mean, it's based on this rather artificial situation that we've created. But the other thing that's increasingly obvious is genetic selection for efficiency and growth is going to continue. I'm kind of sure about that. We're always being asked to produce more from less because of the overall, well, basically the increasing population at the bottom of it. But I certainly strongly feel that we shouldn't make things any worse. We have hopefully a better understanding, although that might be questionable in the case of the problems here. And we shouldn't compound the welfare issues that we already know exist. So what can we do about it? Well, there seems to be two things, at least that we've been involved in. One, and we spent many years, and Paul Hocking, who worked at Roslin, spent a large part of his life trying to see if we could disconnect the reproductive consequences from those of growth. I would argue we were not successful in that at all. Of course, it's not just the reproductive consequences that why birds are food restricted. They're not really very viable if they're left to eat ad libitum. And the other thing which has come more recently, I mean, it's not a new concept, but that in my involvement in it is to look more closely at dietary dilution or dietary composition to improve the welfare of the broiler breeder hen. So, as I say, what have we achieved over, well, the first paper I published, actually, it was with Paul Hocking, on broiler breeders was in 1986. So, and it was in the days when at the Institute you were told, we worked at a government-funded institute, we were kind of told what to work on. So it wasn't necessarily something I had chosen to work on, but it was just looking at how, in this case, dietary fat, just as a way of increasing intake of energy and so on, affected the photoperiod response. And these were dwarf broilers that we could keep in cages, believe it or not. But, you know, we did a lot of work, but I feel we didn't achieve a lot. As I said, we have not been able to find any way of disconnecting the consequences of growth on the reproductive axis. And I can't, in terms of follicular recruitment, so that ovary with a lot of follicles, I'm not sure we have really anything. It's been very hard to find genetic loci responsible for that, per se. But for sure, if you look in, and this has been published by Paul, alleles that come from the broiler type genotype are associated with both heavier body weights and early age onset of lay. That's if you allow them to feed ad libitum, the population. So these are F2 type studies or even advanced intercross. And the genetic loci for growth and puberty are common. And so body weight, as you may well know, is an important determinant for the attainment of sexual maturity. And although this is a concept that's actually been around in the human world literature for a long time, there's very few, if almost any, examples where you can actually demonstrate that. So although it will never make a citation classic, arguably, I think this is one of the few occasions where you can actually see that. So it's not to say that our labours have been completely in vain. What we certainly know is how feed restriction, at least at one level in the brain, reduces the expression of GnRH and reduces LH secretion and results in this more viable ovary. So this work, which Noreen Joseph, who I worked with, and one of the reasons I know Gregory so well, is did some of this work. But as I say, it's not the same as knowing what the genetic consequences are of that gives us this ovary if you don't feed restrict. So if we go back to this as a possible approach, which has certainly been used, and Vicky Sandelin certainly did some nice work and got models where you could get birds on the same growth trajectory I think Vicky, well, certainly came from this stable here. But in our mind, there was always the question, does it actually reduce the feeling of hunger in these birds? Because at the end of the day, they're not getting any more nutrition. They may self-control their feeding, but we argued that as far as their brain was concerned, they would probably still see this as as at least metabolic hunger, even if they're not actively looking for food all the time. So the DEFRA, which at that time, or still does, interested in animal welfare and policy around there, actually challenged us to see if it really mattered that these birds were hungry. So I guess what I haven't mentioned is that broiler breeders, when they're feed restricted, spend a vast amount of time in foraging behaviour, looking for food. They show all the symptoms of being hungry in terms of tests for motivation and so on. But my area was definitely not behaviour. It was to try and look in the brain to see if we could find measures and see if some of these diets actually were satiating in the true sense, at least, in the sense of, in their brain, did they perceive the diets as satiating? We were also challenged with trying to find if there were any negative consequences in terms of behaviour, and that's a lot of work's been done with SRUC in AIR on this topic. I'm actually just going to keep on the neuroendocrine type aspect, but hopefully there'll be more results coming out on the behavioural side. So, as I've said, we're looking at the central control of feed intake, and there's two major players, agouti-related protein and pro-opiomelanocortin. These are genes that are in neurons in this infundibular nucleus in the brain at the bottom here. It's also known as the arcuate nucleus in mammals, and it controls metabolism and ultimately food intake. It does that by acting through the alpha-MSH product, which interacts with this receptor, the melanocortin receptor, or one of them, or two, well, actually two of them, mainly, and to decrease feed intake if you have access, you're fully fed. So that axis is in this way active when you're fully fed or satiated. If you have a situation where food is at a premium or you're food restricted, the AGRP side of the axis increases and actually is thought to antagonize the alpha-MSH at the receptor, and therefore takes off the negative brake and allows you to feed. So this is relatively well worked out, certainly in mammals, excuse me, but in birds, as far as we can tell, the situation is pretty similar. So we chose to look at the expression of these genes in this area in the brain. I'll mostly show you AGRP because POMC just does not actually seem to change very much in any of the manipulations we do. It's always AGRP that's changing. So to try and get some idea exactly how this was acting in birds, we used broiler breeders which were reared up to 6 weeks of age on the classic standard restriction, which around that time is probably around 25% of what they would choose to eat. That's the commercial restricted diet. From then on, we had groups that were on restricted feed, i.e. keeping restricted, released to ad libitum, an intermediate restriction, and also released at different times. So here at around 10 weeks of age, because of previous experiments, we knew they would come to around the same weight as the 2 times restricted. We also released birds, actually not then, but there, 2 days before And this increase in body weight is almost entirely explained by the amount of food that's in their crop, because they just gorge on the food available. So we have a situation with birds at effectively the same weight, here and here, but experiencing different access to food. So ad lib, restricted, of a bit higher restriction than here, and ad libitum. So what were the results in terms of AGRP? Well, having worked on reproduction for a long time, when you're lucky if you can see a 0.5 of an increase or something. Here we have birds on ad libitum feeding, but we did measure them, that's just tiny. The ad libitum birds, there's a huge difference in the expression of this orexigenic peptide in the brain, or the gene for the peptide that codes the peptide. So it's a very dramatic effect that you would expect would be driving the bird to want to eat and change its metabolism to conserve energy. If you look at the comparison where the birds are the same body weight but have different experiences, there is indeed an effect. And this is what actually initially quite excited us, because we thought, yeah, okay, so these birds, we can use this to tell that there is some difference in the way that they're perceiving their current situation in terms of access to food. So it's, if you like, always lower when they have ad libitum access to food, but their body weight is the same. And this is a big problem, of course, if you're looking at birds and if at different ages or different body weights, how do you make the comparison? So we were quite happy with this. But what we had maybe kind of been, by our excitement, a bit less took note of was the fact that the birds that had been released from restriction for different lengths of time also had very big differences in the expression of AGRP in the brain. So if you'd been released for 2 days, it was relatively high. If it had been 2 weeks, it was considerably lower, and that was still statistically different than if you'd been released from 6 weeks. So maybe if we'd taken more attention to that, we would have understood that the rest of our attempts might not be so rewarding. So if you start looking at dilution of the diet as a approach, so-called qualitative restriction instead of reducing just the quantity that they eat. They take a long time to eat the diet, of course, and I'm very careful because I'm not the behaviourist or the animal welfare person, but they seem to show not some of the behaviours that are associated with quantitatively restricted hens. And that can maybe be seen quite clearly, at least here. This is the qualitatively restricted group, and this is a quantitatively restricted group. The litter quality is hugely different because these are drinking a lot of water. So there is at least some obvious beneficial effects of these type of approaches, but we were interested in what was going on in the brain as far as the metabolic center was concerned. I'm sorry I've changed the style of the graphs here, but this is the weight of the bird. So here's a group on ad libitum feeding, a group with 20% oat hulls, so still a quantitative restriction, but not as large. Here's a group with 40% oat hulls, and that's effectively ad libitum feeding. And then the traditional restricted group. So I guess, first of all, you can maintain birds at the right body weight with these treatments, but the bird's not fooled. There's no difference in terms of the AGRP expression in the brain. That system that's going to drive it to, at least we would classically believe, to eat food. We used another approach, a very short-term one. It uses something called Aspergilla husk. It's sold, I don't know in Canada if it's sold, it's used as a to help with constipation, but also with people with problems in their gastrointestinal tract, and I think by bodybuilders to, if they're taking liquid diets to get the perfect body, they use this to bulk out their... Anyway, I'm not quite sure exactly what they do, but anyway, it's sold in massive tubs, so and it's a plant product. In fact, actually, I think it does grow in the UK, but it's mostly sourced from Pakistan and so on. Anyway, the point is it absorbs a huge amount of water. They eat it. You only have to add a tiny amount extra to the diet, and it will effectively make them control their intake. And you can see the crop content. I think we added 15% to the diet that they would normally eat. So not very much in terms of quantity, but it fills a crop. And you can actually see here the crop contents are quite jelly-like. That's a normally restricted bird. That's an ad libitum bird, and this is the Ispagula fed. I think this is in the ileal loop. So again, I think I've already, the results there. It had no, again, the birds are not fooled. They're controlling their intake in the sense to a level that we would like, but AGRP levels are as high as in the quantitatively restricted birds. So, what does this mean? Well, I mean, this is what we expected. I think some of the people that put forward the restriction thought otherwise, but we assumed that this was fulfilled the hypothesis. We thought this would, the result is what we expected. So these strategies certainly do not change the bird's central perception of energy balance. And it's also, to some extent, along with the fact that there was that difference in how long the bird had been released from restriction, there were still differences there, even though all those birds were ad libitum fed. It was beginning to make us think differently about AGRP as a potential marker of satiety. And of course, it comes down to what is satiety. Well, one definition, or perhaps the main definition, is beyond capacity. So I suppose in a way you could say these birds are beyond capacity. They're not going to eat any more of these strange diets we give them, but they are clearly not beyond capacity in terms of their metabolic demand. So I certainly don't think this means that qualitative restriction strategies are not worth pursuing. And indeed, with Aviogen, we're looking at diets where the constituents are altered to try and derive ad libitum feeding, but with more practical diets that can be formulated on a large scale. But we're not really, well, we don't have any results from that yet. But it may not change things centrally, but maybe it's worth it because of the easier management and so on. So, as I say, this made us begin to think about AGRP a bit differently, because at least in the mammalian literature, it's very much, if you've got high AGRP, you're going to see an increase in food intake. But we started to look at situations where birds lose weight naturally. And I need to be really careful because I know this has upset a few people a few times. When I talk about natural anorexia, I'm simply meaning a situation where the animal chooses not to eat and actually loses weight as part of its natural, usually, or at least in the response to some stimuli, reduces feed intake. So I'm not making any hopefully contentious statements about the problems of human anorexia. So we wanted to know if AGRP would behave as we expected if it was truly reflecting satiety. And what we expected was that we would, when birds lost weight naturally, because they were defending a lower body weight, we would not see a change in AGRP. It would stay the same. Yep. But that was definitely not the case. So in this particular sort of model, if you like, these are laying hens. So slightly going away from the broiler breeder. If you move them from cages to pens, there's obviously a relocation stress. They stop eating, they lose some body weight, and actually they'll also stop laying often. I'm sure it would work the other way around. It's no particular statement about cages or pens, it's just a new environment. So feed intake goes down. This is over a short period, I think 4 days, but there was a large increase in AGRP. So these birds are choosing not to eat, if that's the right word. It probably isn't in a behaviourist sense, but nonetheless, AGRP is going up, so that signal should be telling them to start eating. And then the other area that, for other work I've done, was very familiar with, is that when birds incubate, when they sit on their eggs for 3 weeks, if they're, obviously most eggs go into a hatchery, but if you let birds do it, they will lose 20 to 25% of their body weight, and they come off occasionally to drink and sometimes eat, but basically after a week or so they're pretty much stop eating. And we pair fed birds to the amount of food that, when a bird went broody, we pair fed another bird, and one was chosen to be a control at the same time, and when these birds were killed, we killed the others. So we called it a yoked control because it was matched, not yoked with a Y, with an L. And so we called that voluntary and involuntary anorexia. And I say, not saying anything about human anorexia there. But again, in fact, if anything, in the voluntary anorexia, AGRP levels were higher than the control. I suspect that is not significantly different when you tested it, but anyway, even if it was, it's in the wrong way. So we had a situation again where the actual observation of feed intake was completely disconnected from the levels of the orexigenic AGRP in the hypothalamus. So one thing, and this was actually why we did this experiment, was that there was this question from the Ministry of Agriculture, or DEFRA as it's called now, to put the level of food restriction that a broiler breeder sees into some context. Now, indeed, these birds that are incubating are actually undergoing, the AGRP elevation is similar when you try and correct for the other factors to what we actually see in a food-restricted broiler breeder. So that was a kind of philosophical point that they wanted to know how bad is the level of food restriction. So that bit of information was fed to them, fed to the DEFRA, not fed to the bird. So the question is, can you really compare one situation with another? I don't know, but that was one of the reasons we did that. So, what though with all the other information, it suggests that AGRP is probably reporting the metabolic status of the bird, or the distance from where it would like to be in terms of its body weight, rather than directly the drive to eat. And there's another obvious thing is, and that might be something in the future that's useful, there's got to be something that's standing in between AGRP and the drive to eat. And relatively recently in mammals, this gene and rather its expression in a specific nucleus, has been shown to be related to when bad things like sickness and so on, AGRP has to go through this sort of gate to have its effect. And if you're suffering from stress, certain stressors, it seems to. So of course we have no idea in birds whether this is the same, but I think this is one way where I'd certainly like to look, because again, maybe that could be used to control feed intake. Maybe. Okay, so I've mentioned set point a little bit, so where the animal might like to be, and the concept of set point is clearly way too simplistic, but it's a nice way to think about things. That you would have if you have a high set point, you're defending a higher body weight, and so when you're satiated, you start to eat less and come back to the middle. Of course, it's a wide band in reality, and if you're dropping in weight, you'll start to feed, and so on. But it is quite a useful way of kind of just thinking about things. So that led us to think, given that AGRP, at least in the bird, was seeming to reflect much more the distance it was from its body weight that it would, if you like, be aiming for, we began to think, well, maybe there'd be genetic differences in the level of AGRP expression. And so the question is, what are the two genetic loci with the largest effects of growth in poultry? And one of them is quite obvious, but I guess people don't always think of it as a genetic effect, and that's sex. And the other one is one that I can't say that we're not the only ones to have discovered it, but we've elucidated it, and that is a major loci on chromosome 4, which is consistently recognized as the largest effect on growth in chickens when you cross any lower growing strain with a fast growing one. And these was the classic layer broiler cross that Paul Hocking set up in Roslin. And that was where we got the who ate all the pies gene name, because this was, of course, it's not the whole story, but it's quite a large bit of the effect. And basically, with a lot of work on genetics, and this is into the 8th generation of crossing between broilers and layers, and we're now in the 23rd generation, we were able to fine map a loci, which when you take the best SNPs, and we added this one later, but you can see here there is a gene called cholecystokinin A receptor. Now, to us, it seemed very obvious that that might be a strong candidate for controlling growth, because cholecystokinin, the ligand for the receptor, which is named, of course, after its effects on the gallbladder, is all over the intestine. It signals to the vagus to go to the nucleus tractus lataris, I think is the right name, where again there are CCK expression, and again there's also A receptor expression, and in the feeding center that we've been talking about. So it seemed a very good candidate, but and also there's conflict in the literature whether it's circulating cholecystokinin coming from the intestine and the other organs, or whether it's all through the vagus, but take your pick. I think it's probably a bit of both. It accounts for approximately a 12% in this generation, but we've seen up to nearly 20% difference between the haplotype, i.e. if we genotype at that locus, well, this is a particular SNP, we're getting up to a 12% difference in body weight, in this case at 20 weeks of age, different ages, different size of effect, but it's reasonably large amount of body weight. So we had to try and work out if it really was cholecystokinin A that was causing the effect. We only had a genetic association, so we used the offspring from heterozygotes, so we can breed them and get all the different genotypes. We sequenced, we looked at giving the ligand on food intake, measured expression, and something called allelic imbalance, which helps you understand how it's being controlled. So, to cut a long story short, if you inject cholecystokinin into the peritoneum, the low-growth birds reduce their feed intake in the 30 minutes after quite considerably. In the high-growth genotype, they're almost refractory to that signal. That was encouraging, to say the least. When we looked at expression, if you look across all the tissues here, there's always the low-growth birds are higher than the high-growth birds. The only exception was, I think, ironically, in the gallbladder, but again, numerically, it's in the right direction. If you look, we raised an antibody expression in the hypothalamus areas. In this case, again, different number of neurons observed, always higher in the low growth. If the factor was maybe something nearby acting through a transcription factor, it would act on both copies of the gene equally. And you would see that if you looked at if the allele, i.e. the gene, had been inherited from the high-growth genotype or the low-growth genotype, you would see no difference in the relative expression of both of them. That was not the case if it's the gene if the gene's acting on its own, or rather the effect is purely at the gene. You'll see an imbalance between whether the bird inherited that allele from the high growth genotype or the low growth genotype. So in a heterozygote, there's a difference between the two copies, which is quite large, about 3.5 times more coming from the low growth allele in terms of expression. And what was perhaps most interesting, although I'm not sure we realized it at the time, but in relation to all the other work, is that when you looked at AGRP expression, in the low growth birds it was low, and in the high growth birds it was high. So purely on this genetic loci, we're seeing a difference in the level of expression of AGRP. Say POMC is in the opposite direction, but not quite significant. Again, POMC seems to have which is anorexigenic, is much less, at least in terms of expression, less dynamic. So, I said the other genetic effect is sex. This is the growth curves of actually birds segregating for the high growth and low growth allele, males and females. So you can clearly see that males, and you probably know this, grow faster than females. This was, again, a bit of an experiment of serendipity due to missexing when we hatched these broiler breeders in our own facility. But this is going back to the Ispagula husk experiment. So we had roughly equal numbers of males and females in that experiment. And we had quite good numbers, fortunately. So I think at least 24 in each group. So there's around about 12 males, 12 females. Always, whatever the treatment, the male has higher levels of AGRP expression than the female. We were so surprised at this, we repeated it just with ad libitum and restricted. And again, same result. The male is always higher than the female in the equivalent dietary group. And perhaps most importantly, in a genetically distinct line and without food restriction, male higher expression than female. So it appears that this could be part of the way that the animal is, that these genetic effects are ultimately being expressed as a difference in body weight and growth. So, to kind of summarize the genetic aspect, we've definitely shown that cholecystokinin A receptor is maybe not beyond all doubt, because we don't have you can't easily knock these genes out, or it's very expensive to do it. We can do that, but it seems to be the functional basis of the QTL for growth on chromosome 4. And again, I would argue we haven't probably proven it fully, but there's good reasons to believe that the mechanism by which it may do that is at least in part through its central effects and probably in the control of the orexigenic AGRP neurons. I'm sure it won't be as simple as that, but that's a sort of hypothesis that we're working towards. And the sex differences in growth that you see could, to some extent, or maybe a large extent, be due to this difference. So, going back to this sort of set point model, if you have a high set point, i.e. when you have the CCK high growth allele and you're a male, you presumably have a higher AGRP level, which of course is still changing in relation to your feeding level, but is set at a higher level. And the converse if you're if you're a female and in the lower growth haplotype for CCKR. So if we come back to what the Ministry of Agriculture really wanted us to do, and is AGRP a marker? We can't say that it always reflects satiety, for sure. It seems, if you extract the data from that first experiment I showed you, this is a major part of its effect. It seems to be reporting and maybe driving the animal towards its correct body weight, because of course as these are released at different times from restriction, they've got different different amount of weight to gain to get up to the body weight that they are genetically, if you like, programmed to have. So that's the increased body weight, and that's the AGRP level. So that these birds are furthest away from their, let's call it their set point, where that one is closer to it. The other implications, I guess, or observations is that this sort of confirms a lot of people's belief, of course, that birds have been selected for changes in appetite during domestication. And this is, I thought, quite interesting. So this is, again, something that Paul Hocking put together. It was what they called the Multiscreen Experiment. So there are however many dots there worth of different strains of birds, and this is their 10-week body weight. And so there's commercial layers in there, there's commercial broilers, but they're because their body weight would be off here somewhere. It looks nicer just with the traditional lines and the layers. But basically, there is quite a good correlation between the frequency of the high growth allele marker in the population and how heavy the bird is at 10 weeks of age. So you can tell these are much bigger because the barn is small and these are the size of a house. But these fowls, these are pictures from, I've forgotten the name of the person, but they were, I think, a book that was given out at the World Poultry Congress in Amsterdam. And, excuse me. So that gave us some hope. Okay, I don't think it's going to get any better. So anyway, domestication, selection for higher body weight. And of course the broilers were all fixed for the higher growth allele, almost. What can we say about the qualitative restriction? It works, but it doesn't change the central feeding center.
So in both the experiments, same result. So it seems unlikely that mechanosensory signals from the gut can actually reduce hypothalamic gene expression. So we need to look for different ways of trying to measure that. I mean, maybe we don't even need to. If it works, it works. So this is maybe where I need the help, other than having a throat lozenge. If the genetic set point for metabolism has been altered, and if you like, the satiety bar lifted, is it wrong to feed restrict them? Because that is the welfare question, is that, you know, these birds are hungry. But on the other hand, they're still growing, and we have, you know, they are healthy. And then the next thing, I think, is can we utilize any of this knowledge to find dietary or genetic solutions to this paradox? And that might actually reduce the central AGR expression. And we are on the cusp of a genetic revolution, and we have the tools now to edit and successfully produce edited animals, but it obviously have to be a sophisticated control. So maybe we can also harness some of these natural anorexias in some way, but I guess I'm a little pessimistic, but I think there is some hope. And so these are all the people I've worked with, particularly Tim in Newcastle. We published most of this work, I think, that you've seen, just in the last month. Paul, who unfortunately is not so well at the moment. Some of the work on the AGRP immunohistochemistry with Simone's group, and Dave Burt involved heavily in all the tools that we could use in the genetics to be able to sort this out. And colleagues, particularly Rick and Laura at SRUC, who have really done a lot of the behavioural work, and who, you know, hopefully sometime you'll see the results of that. But I wouldn't be able to do that justice. But it's been a very fruitful collaboration with the welfare and the neuroendocrinology. So sorry for my wassail voice, but thank you for, well, coming and listening. Thank you.
Well, thank you for that great presentation. It actually answers the two questions that I had. Okay. We have time for questions from the audience, but as I said, we've got...
In sign language.
You feel a little bit of one questions just now, and then we can come forward and...
No, I really would, because I know there are people here working on welfare and all these sort of things. Are certainly, you know, I think the broiler breeder problem is going to be with us for a very long time. But, yeah, so I would be more than happy to hear any ideas. Or if we can help in some way.
Maybe I'll start with one question. When you look at the cholecystokinin message from the digestive tract, what is it that triggers it? Is it just the physical distensions or the physical presence, or is there specific nutrients or metabolite that are responsible for activating the CC peptide?
Well, ironically, and this is, of course, one of the puzzles, we have not seen changes in cholecystokinin expression in response to any of these dietary treatments. I would say at the moment, the thing that's having the largest effect, the classic satiety factors that have the largest effect seem to be PYY. So, of course, you're rightly asking, how does this mediate? Clearly there is CCK expression, and it's, as I say, I think for this, the critical part is in the brain. We've measured gut transit time, food conversion. It seems to be efficiency, not more food intake in the act. Okay, you inject CCK and you see a difference between the genotypes, but when you actually look at normal food intake, we can't see a difference at the moment. A difference in food conversion rate, efficiency, just about. A little bit difference in the gallbladder size, but I think, arguably the biggest effects we've seen are in the brain.
My concern is like when you use a diluted type of diet, the actual amount of energy and nutrients that enters the blood is probably similar to the restricted eating.
Although over a longer period, of course, so we do see differences over 24 hours in some of the gut parameters, but I think it's because they're being, the absorption seems to be taking place over a much longer period, I think. So we have got data, we've got some really big experiments that were done looking at food restriction, ad libitum fed, the oat hull things with different time periods, and nothing's particularly stellar there, but one or two of the factors that respond to feeding in terms of like PYY change quite a bit, and also you can change their profile by the dietary means, I think, but we're not making those. It's not as if the diet, these diluted diets, make them show any signs that they're signaling more. In fact, quite the opposite. I guess you've probably just got an a hungrier bird for longer, as opposed to it at least having a period where it's satiated because it's had its fill in the morning. I mean, that's me. I'm not sure we're improving the welfare from that point of view.
Well, I think my thing is like, is it better to feel hungry or not feel hungry but still being malnourished? So, the difference, and I don't know, maybe the welfare can answer that. If you put a filler that makes you feel full, but you're still malnourished, is it acceptable?
That's not a lot. So you're not going to get it. You can try and feel pain or have it too often. That would mean malnourishment unless you're feeling bad, feeling poorly, not a welfare. Others would argue so much. That's pretty much from the viewpoint.
That's kind of.
So trying to figure out the feelings gene is more important, because a lot of times when you go on like the keto diets, they feel most. So you're losing weight and not feeling hungry. So if you can figure out what are the mechanisms for losing weight and not feeling hungry.
That's what these diets were appraising from the industry were doing. They're adjusting the ratios of nutrients to see if they can achieve something in that way. Yeah. So that'll be really interesting. And of course, what DEFRA, and of course I've not mentioned it because it was absolutely nothing really, but because we saw no effects. But what they challenged us to do, as I say, was I think it was appraise the subjective state of the broiler breeder. I thought I would never work on anything like that, but of course, so we were looking at markers of depression, IL-6 in the hippocampus, hippocampal volume, God, I don't know, not a sausage to use a...
Because when you mention looking for natural feeling like this takes mind. But yeah, is it just that there's no endocrine status is very, very different? Is that change actually suppressing and feeling the hunger?
Yeah, exactly. There's got to be something. High prolactin is the obvious thing, and it must be in
well, it doesn't have to be the high prolactin. It could be the VIP neurons in the brain that are projecting to, let's say, these calcitonin gene-related peptide or another circuit that must be intervening. But the fact is, yeah, so, and this is what made us reappraise this AGRP, because I think there is a general dogma that, if you've got high AGRP, and it's very true, if you stimulate those neurons in a mouse, they will immediately start to eat. But of course, in these situations, it's high. We're assuming that's a marker of the activation of those neurons, and they're not eating. So there's got to be something that's preventing that signal acting. And it is interesting. I mean, how you utilize that, hard to say. But at the end of the day, I guess, we shouldn't stop looking because, as we know, you never know.
Well, in the mouse study, so you stimulate those neurons and then it's eating. All else is the same at that point. So that's the only thing that you're really.
Yeah, it's incredibly precise.
A whole bunch of stuff, right?
Of course.
Yeah.
No, it's not as genetic. It was the best we could come up with, and I think, to be honest, that's probably why we got the grant, is because we had something that at least tested the hypothesis. And I think Rick, who obviously is much more the behavioural person, I think maybe he's changed his mind a bit. I mean, I think he's a bit more, seems a bit more conducive to the food restriction. Maybe isn't such a bad thing in this situation. But then, of course, it is a paradox, because if you didn't, they'd be incredibly unhealthy.
With the selection for the broilers, so it's not only their high appetite, but increases. Some of that maybe changes in the diagnosis system.
Of course.
Behavioral as well, right? So
yeah, which I wouldn't be surprised if the AGRP, because it does change activity. It changes the way the animal behaves. So more energy-saving strategies. So it could well be at the core of all that.
I'm thinking about studies that
Oh, sorry, no, but wait a minute. It's entirely consistent with that. Yeah, no, that's right.
And like perianthum, someone like when they compare, no falls or like high octane nitrogen, and the amount of conscious preloading that they do, and so conscious preloading.
I've certainly heard about it. Yes.
Free food versus having them work for it. And the red jungle fowl will choose to work for 7%. The modern laying hen will only work for 3%. She'll take 70%. So we've changed, with the selection, there's something that's different about just they're behaving more. They're not doing things to waste energy.
Yeah, no, absolutely, yes,
right?
yes.
And there's probably a whole bunch of ways that that's happening.
Yeah, and as you say, obviously, you can have a higher, I think theoretically you can have a higher drive to eat. But if you don't then utilize that food efficiently, you're not going to grow it. So, the broiler is doing all these things. It's got, we know it's got differences in some of the amino acid pathways, and that will make them more efficient. So there must be thousands of those. Of course, it's a but this just happens to be the largest single effect as far as we know. But, yeah, obviously I suppose the whole process of domestication is to drive, to change things to suit what we want, and if I suppose behaviour is a huge part of that.
Well, they get like reduction in fearfulness is energy saving. So you look at, and then cattle look at people measure fearfulness and very much.
There's good genetic correlation in laying hens, and I think in broilers as well, between ranging ability and their output. So, the ones that lay more are less, whatever you want to call it, I can't remember, on that sort of fearful. They're the ones that don't go out. They're not exploring novel. So unfortunately, although people would like them to go out, the selection is probably working against it. And I think they showed the same with broilers. Somebody in Australia, I think, has done that.
Well, those who waste energy are not get blood.
Yeah, of course.
Right, so it's interesting because people want to see if it kind of, people feel better, animals are performing energy-based nutrition.
Yep.
Whether the animals are motivated to do that or not.
Then we should all be ready to pay more for the energy waste.
Right.
All right, on that note, it's the 2:30, so I know some of you probably have other things that are planned for the day, but as I said, we've got the room for another hour or so if needed, so feel free to stick around and have a chat with Ian and for the others. So thank you all for coming. I hope you enjoyed it, and have a nice remainder of your afternoon. All right, then, thank you, Ed.